I'm not really used to this open format with the rock star kind of mic. So, where's the pointer? Thank you. Thank you. So, yes, what I'm going to talk to you about today, and following on from what John said, is I'm going to delve a little bit more into B-cell immunobiology. But first, I would like to thank everyone, the organizers, and I know I've got a picture of Esper up here, but I'd like to thank everyone, including all the sound guys and the people in the back there, who organize and put on this course every year. And from my perspective, it's getting better and better, and the posters the other day were wonderful. So thank you very much, and well done to all of you. So, onward. So, I used to be a T-cell immunobiologist with some of the characters who are in this room. And when I got to the Vaccine Research Center, I began to study these strange things called memory B cells. Now, memory B cells, while they are lymphocytes, they actually differentiate into these Golgi factories. These cells are amazing at just producing protein. And how the cell gets from being a circulating memory B cell or a naive B cell and get to being a plasma cell, a long-term plasma cell that resides in the bone marrow, is a fascinating feature of immunobiology. So I shan't tell you all about that, but I just think it's fascinating. So, at the Vaccine Research Center, we use two diseases, or two proteins, to study memory B cells. We use influenza HA, and also John did an excellent introduction about this EOD, Engineered Outer Domain, germline targeting 8, version 8. And he did a great introduction to that. I'm going to talk to you a little bit more about how we do sequence analysis of specific lineages of those B cells. Then I'm going to talk to you about the Liverpool football team. No, then I'm going to talk to you about the diversity of some of these sequences in different populations. And I use this because there's an Egyptian, someone from Senegal, someone from England, someone from Turkey via Germany, and this chap Firmino, who's from Brazil. And then I'll move on and talk to you about some evolving B-cell phenotype analyses that we have going in the lab. We're trying to pull apart some of the traditional ideas that we have about how we define memory B cells. So a bit of basic biology first, so we're on the same page. This is my version of a dynamic germinal center. And in this germinal center, particularly in the light zone of the germinal center, this is where these three friends all interact. And the friends are the B cells, the follicular dendritic cells, and the CD4 T follicular helper cells. Now, they all get together, and if you don't get any attention, the B cell then dies. However, with signals and antigen presentation from the follicular dendritic cells, the B cells go into the dark zone. And this is where all the proliferation and the somatic hypermutation takes place. For reasons or for signals that we don't quite understand, from the germinal centers are released short and long-lived plasma cells, which then go into the bloodstream and they produce antibody that we can detect by a simple ELISA. Indeed, some of these plasma cells will traffic to the bone marrow, where they will take up occupancy in the bone marrow and keep you producing antibody for the rest of your life. For example, measles, mumps, rubella. You only need to be vaccinated once. Also, for signals that we don't quite understand, memory B cells are liberated from these germinal centers. And these memory B cells are far more diverse than actually the antibody population, because these are waiting for an antigen to come along and then differentiate into plasma cells. that produce antibody and protect us from different diseases. So this is a very heterogeneous population with a greater diversity than even these antibodies. And we call this the recall pool. And I'll talk a little bit more about this. So upon encounter with antigen, some of these high affinity memory B cells will traffic back into this germinal center and lead to a different immune response. So today I'm going to concentrate just on these memory B cells. If we have a look at all our immune responses, we get exposed to everything. Okay, so any antigen that we may come across, for better or for worse, we tend to generate an immune response. But we also come across vaccines, and this is intended to elicit a specific immune response. Now, these immune responses can be individual antibody repertoires, so each of us may have a different population of memory B cells, or slightly different antibodies that all elicit protection. And these are reflected in your recall repertoire, in your memory B cells that are in your circulation. However, more often than not, and some of the antibodies that Dr. Muscola talked about just this morning are common recall repertoires. So in other words, do we all have the ability to make a VRC01? Do we all have an ability to make antibodies against a specific epitope on head? Well, yes, we do. But that's all residing in our repertoire. So, this is, have you seen this a few times already today? This is the spike of HIV, and these are highlighted on this are the different areas that Dr. Muscola talked about. And today I'm going to concentrate on the CD4 binding site, particularly this VRC01 lineage and also VRC07 lineage. One of the things that we find very difficult about generating or eliciting or inducing antibodies against HIV is this problem that they are so highly mutated, up to like 31%. So can naive B-cell immunogens, such as EOD-GT8, and sequential vaccine strategies that Dr. Muscola went into detail, can they induce high rates of somatic hypermutation? But there are some considerations. These are not all the considerations. These are just the ones that I think about. Is your HIV envelope immunogen, is it manufacturable? So can we reproduce that all the time? And does it engage the distinct naive B-cell lineage that we need to elicit our antibodies? Is that lineage and the engagement of that lineage, is that predictable? And are common signatures? Is the elicitation detectable as a neutralizing antibody response, or does it just reside in that memory B-cell pool that I talked about? Can we easily identify the B-cell receptors of the intermediates? And then what's the influence of the B-cell phenotypes? So for example, not all of us can be, think of a president or a footballer. Not all of us can be Ronaldo. But how did actually Ronaldo start off life? What was his education when he started off playing soccer to now being one of the world's best soccer players? Or the best soccer player, depending on where you're from. Clinical trial rigor. This is something that I've got more and more into. As we go into the clinic with these kind of immunogens, we have to have reliable tests to tell us that that particular immunogen is working. And so this is the currency of this clinical trial rigor, is specificity, sensitivity, how robust is it, and therefore what's the reproducibility. These are just some of the planned HIV trimer clinical trials that are ongoing. And we see here there's the EOD-GT8, and that's due to start next month. We'll start recruiting for that. Then there's this BG505. BG505 is a trimer, so it's a native trimer. And again, BG505 here. But what Rohir Sanders and John Moore have done, have reverted some of the neutralizing antibody sites that we know back to those that would engage naive B cells. Also have BG505 here, and of course then we have one at the VRC as well. So these two clinical trials are going to require as a primary readout the B-cell intermediate analyses that I'm going to tell you about. And then the secondary readout would be HIV neutralization. And this is very unusual that this is a primary readout. So I'll tell you more about the EOD-GT8. This is the clinical trial that starts next month. The clinical sites are in Washington, D.C. and also in Seattle. It involves 18 individuals in 2 arms that are separated by dose. So it's a low dose and a higher dose. As Dr. Muscola explained, the EOD-GT8 is displayed for the purpose of generating a reliable immune response on this lumazine synthase, which gives it a valency of 60. There is a control here as well. And then, of course, we have the adjuvant, which is a GSK adjuvant called AS01B. So what are we trying to do here? So here's our favorite lumazine synthase, 60mer. And here we have the specific naive B cells bearing this heavy chain that we want to engage. These go into our germinal center again, and out of the germinal center becomes those antibody secreting cells, and also the mutated memory B cells that have been engaged and then differentiated into memory B cells. So we've taken that. We then come back with variant number 2, which is actually designed upon the results of the B cell intermediate. So that's why we have to be so precise about our B cell analyses. Once again, they go into a germinal center, and we have mutated memory B cell number 2, and also we have those antibody secreting cells. This process may be undertaken to drive somatic hypermutation sufficient to achieve neutralization. And also, can we selectively identify phenotypic and genetic differences between this initial memory B cell and this B cell? So this is a table of all the antibodies that have currently been identified that recognize the CD4 binding site of HIV. And as we can see, there's a whole, what we call a VRC01 class, which involve all these guys. And some of these have got different potencies. For example, this is VRC01, which has a potency of 0.5 milligrams per mil. But then we have this guy N6, which has 10 times more potency. So one of the questions that we asked ourselves is, when we use these immunogens, which antibody lineages can we drive with the vaccination? Notice as well the percentage mutation rates that these have, both in the heavy chain and the light chain. But also notice here that there's a very defined heavy chain that we can define from the memory B cells when we do the analyses. So how do we do those analyses? This multicolored bunch of dots basically represents all your circulating naive and memory B cells. And what we want to do when we add EOD-GT8, we want to engage some of those, not all of them, just some of them, to drive this lineage. How do we tell that we've engaged these B cells and they're in the periphery? Well, we use flow cytometry and a thing called index sorting. Flow cytometry, in this case, we have a whole panel of markers that will identify memory B cells. We then use these 2 probes, as we call them. Some people call them tetramers. For EOD-GT8, as it appears on the surface, and this EOD-GT8 variant. And what the variant is a knockout in some crucial residues that the antibody would recognize. And so, using flow cytometry, we can say, well, these guys that recognize the variant and the native protein equally, we probably don't want those. But what we do, we want the ones that don't recognize the variant, but recognize the EOD-GT8. Okay, so the assessment of these, we're going to use, in the clinical trial, we're going to use binding, ELISA, octet, or neutralization. But also we're going to sort those cells and put them individually. So every cell gets its own well, and then we do PCR on those wells. So what do we expect? Neutralization of HIV is not expected from the prime. Okay, so the EOD GT8, as it stands, will not elicit neutralizing antibodies. Binding assays, such as ELISA, will only show us overall immunogenicity. So if I punch you on the arm, how loud do you say, ow, right? Then we can assess, but this is the key point, we can assess the frequency of the immunoglobulin gene signatures from the VRC01 class B cells. So what we're looking for are those B cells that recognize the protein that are VH1-2, we'll also take the -3s, etc. So that's theory. And this is a bit more in detail how we actually do the panel. We have other markers in the panel, and then every single cell, based on this gating scheme, every single cell we can go back to. And index sorting means we can tell what every single cell was bound to. So we can know what phenotypic characteristics of every single cell. We then do rounds of heavy chain or 2 light chains, the kappa and the lambda chain. And currently, this workflow is for Sanger sequencing. But we are working with Dr. Dueck on trying to get whole immunoglobulin sequencing high throughput. And as a bioinformatician, you could probably appreciate that, right? So what else do we have to consider when we identify these EOD-specific naive B cells, particularly when we're doing it for a clinical trial? Well, with Dr. Kaub, we immortalized the VRC01 class naive B cell as a positive control. And that was a tremendous reagent, and it really pushed us forward. We tested, in this paper that originally described the broadly neutralizing antibody, they used just 2 individuals and got a frequency from 2 individuals. So we wanted to be sure that we could actually detect the frequency of these kinds of immunogens. So we tested 50 volunteers. Then also we said to ourselves, well, your target population isn't actually just in the USA. So we took some populations from South Africa, from Uganda, and from Rwanda. And we found some pretty interesting things. This is just to show that, this is by Alberto, who's in my lab. And what he did here was an absolute tour de force of persistence. He spiked into PBMCs, immortalized VRC01 class B cells. And he did this by first separating the immortalized cells using flow cytometry. He then used the cell sorter as a liquid handler and put maybe 1 cell or 2 cells or 3 cells in a well, and then processed all the cells and see what he could get back. And what he got back was, so he did this 3 times, and his precision was really quite good. So he knew how many VRC01 class naive B cells there were in each well, and this is the expected frequency versus what he got back. And of course, he shows off that he gets 0.99995, right? So basically, we got sensitivity down to one naive or memory B cell in 5 million. So this assay is highly reproducible. So then he took populations of white Caucasians from USA, African Americans, and Asians, all resident in the USA. We then looked at South Africans, Ugandans, and also those from Rwanda. And what we found when we're looking at naive B cells, we actually found there was a difference in the circulating naive pool that was recognizing EOD-GT8. So on average, there's about 1 in a million naive B cells could give rise to the VRC01 class broadly neutralizing antibodies in individuals who are not infected. There's higher frequencies of VRC01 class naive B cells in individuals living in South and East Africa. And that's what we're chasing right now. We don't know why that is. And maybe in different populations, maybe in a Brazilian population, there may be a different frequency of naive B cells specific for different antigens, which we don't know. It could be associated with antigen burden. So these individuals are clearly living a different lifestyle to these individuals. And then we analyzed all these sequences for the presence of, remember that table I had? For the presence of all those other antibodies. And this is difficult to see, but basically look at the table. So of all the individuals that we looked at 1,712 memory B cells. We found that the predominant signature that we got was VRC01. But we also got N6. We also got IOMA. We also got this 8ANC131 and etc. When you look at the non-African, those individuals that are resident in the US versus those that are resident in Africa, we also see there's a difference in the distribution of naive B cells for those different antibodies. We also looked at memory B cells because we could, essentially, but that produced something that was very interesting, and we found a few VRC01 and IOMA-like memory B cells could be identified in those individuals in South and East Africa. It's not tremendous amounts, but that shows that clearly antigen burden could cause some problems in mimicry of this protein. So maybe they've seen elements of this protein before. This is pardon me for this slide, it's very faint, but essentially this just shows us that there's more IOMA and VRC01, or there are actually the presence of IOMA and VRC01 memory B cells in the African population and none in the American population. So we're expanding this study and looking at consistent trends. So what we found is the naive B cells exist that recognize the EOD GT8, but they're not just confined to the VRC01 lineage. And interestingly, they actually can be this N6, which is incredibly potent. So we have to cast our net slightly wider when we're doing the analysis of the clinical trial. So just in conclusion, we've developed a robust, specific, and reproducible, and it's precise, workflow for B-cell lineage analyses, good enough to run a clinical trial, which starts next month. We got higher frequencies of both naive and memory B-cells, at least the VRC01 and IOMA class in memory B-cells that are measured in different individuals in South and East Africa. Those proportions change depending on which antibody lineage is engaged, so VRC01, N6, IOMA, HJ16, and VRC16. And also we identified a few pre-existing memory B cells, which we're following up as well. So I will now move on to some more B cell analyses where we're looking at phenotypes of B cells in vaccination. So because we have these rather smashing machines, courtesy of Dr. Mario Roederer, who is one of our colleagues at the Vaccine Research Center. These, for those of you familiar with flow cytometry, these have 5 lasers and can do up to 50 parameters, which practically means you can probably do routinely 20 to 25 color flow cytometric experiments. And then you say, well, there's not that many dyes. Well, there's this country, Sirigen, this company, and they're developing more and more dyes. So it could get up to 30 colors routinely. Now, I'm going to switch gears and talk about flu just for a very short period of time. So, immune history, particularly in influenza, and this could be something we need to consider for HIV vaccines at some point. So in my 50 plus years of life on this earth, I've been exposed to different influenzas. And so there's me in my pushchair in 1965, and I've probably been exposed to H3, H2, a circulating H1N1, group 1, a very strange lab accident at Rockefeller University, so I probably got 1918, group 1, and also pandemic H1N1. Now, my children were born in 2009, and they will probably have a completely different history, having only been exposed to perhaps pandemic H1N1 and H3N2. So what does, immunologically, what's going on here? Well, every time you see a drifted head, you encounter a new B cell, right? So new B cells are getting recruited that recognize head. The stem, meanwhile, is rather like a trunk of a tree, and it doesn't change very much. It does have some glycosylation sites on it, but in general, you recognize the stem consistently, be it a group 1 or a group 2 stem, but you recognize that consistently, which makes it a nice target for a universal influenza vaccine. So, you see many of these probably when you were doing undergrad. So this is flu number 1, this is your primary immune response. You then get secondary exposure by flu number 2, and those primary immune responses against common epitopes increase. However, those against epitopes that are new because of drift are brand new. So can we have a look at those B cells versus those B cells and try and see what the difference is between the new ones and the old ones? And of course, this has lots of application to looking at these lineage vaccine trials, and one of the reasons why we started off doing it. So this is H3N2. This is the Victoria strain. This is not the nasty one that has just caused lots of illnesses in the US. And this is a clinical trial that we did at the Vaccine Research Center using another group 2 flu called H7N9. And those colored red here, those residues colored red, are the ones that are different between this flu and that flu. So this makes a very good model at looking at B cells that recognize new and existing or old. And we do this by flow cytometry. Clearly, we've all seen H3, but we've not seen H7 yet. 2 weeks post-vaccination with this H7, the H3 go down actually, and then we have a new H7 population. Now, traditionally, we have looked at memory B cells, and I presented this some years ago, based on CD21 and CD27. And you get resting memory B cells and activated memory B cells. You also get this other population, which is called atypical, exhausted, or tissue-like memory. And because it's a negative population, you can make up your own name for it, because really, you're not positively selecting it. This has been described in the context of both in malaria and also by Dr. Fauci and Susan Moore in the context of HIV. And these cells are very interesting in the context of HIV. But if we look at those, if we look at IgA, HA memory B cells, what we see at pre-boost, you have a lot of resting memory B cells, and they're probably the ones that are recognizing the existing epitopes. Then at day 14, you get more of these activated. So the cells are waking up now, and they're going into germinal centers, and they're becoming more activated. And then on day 90, then that activation's gone, and you're back to this resting phenotype. We also know, and this slide summarizes a lot of work. So resting and activated memory B cells are transcriptionally distinct. So here, for example, the ones in red are activated and the ones in blue are resting. This was done using fluidigm, which is a very selective technique using very selective probes. But this was done using single-cell transcriptomics. And what we see is that the blue and the red cells tend to dissipate using a PCA plot. So we reckon that there's 155 genes in common that differentiate resting memory and activated memory. Using this and then going back, selecting the cells, sorting the cells, doing transcriptome again, we actually come to, we did this 2,000 times actually. Oh, sorry, we did this on 2,000 cells, which is essentially 2,000 times. And what we found, we begin to find that activated memory begins to drop out into these different groups. And this is using things like CD85J. And for those of you who know the literature, this is associated with Tibet, FcRL5, which is inhibitory, CD19, of course, CD72, another inhibitory, et cetera, But we had more than 25 markers, of which we developed a panel using 21 markers. So then we start to use these multi-dimensional analyses using this thing called TISNE. And if we have a look at these are the pre-existing stem-like cells, the H7H3 cells. And as you see it boost, they're in this kind of quadrant here. And then at day 14, they're becoming more activated. And then they go back to their resting state a year later. However, for the new ones, remember, these people have never seen H7 before. So in the new ones, we have some cross-reactive cells for H7. They kind of sit in this quadrant at day 0. But then look at this. All these cells have they're all going in different directions, not necessarily just going down one path. So it's not like memory T cells. All these B cells are going down different directions. And this is using a lot of data from the transcriptomics. Then once we get to a year, they're still kind of dispersed. And they're going back to where they were before. But still, you've got some that look rather like these guys. So we're doing a lot of work right now trying to differentiate more and more what's going on with these memory B cells. Are memory B cells all created equal? Well, if we start to look at the lineages, we know that they're not only just phenotypically distinct, but also they derive from related lineages. And we're doing a lot of work now expressing some of these lineages as antibodies and seeing if they have different neutralization potential. So maybe one of these activated phenotypes is more, what's I'm looking for? So maybe there's more neutralization in a certain activated phenotype, which would probably be really good for immune monitoring if we could say, particularly in the EOD trial, if we could say, if you have that phenotypic B cell, therefore you probably are on the way to neutralizing antibodies. So what I've just described to you is that you have these different clusters. These different clusters, we can put them back onto CD21 and CD27. And really, the resting is probably an homogeneous population, but then you have this immature memory phenotype, and then you have all of these, which are kind of interdispersed. So we're on the way. We're not there yet, but we're on the way. So maybe I'll stand here another 2 years' time and say, well, we've got a memory phenotype that elicits neutralizing antibodies, but I'm not sure yet. However, if we just look over time, these H7 new cells, if you have a look at AM3, activated memory 3, 2, and 1, and that's what we group them in for now, compared to the pre-existing cells, we see that there's more AM2 in this population and less in this population, and probably more AM3. So we can make some sweeping statements about these populations. Like AM2 is associated with a more established memory B cell response, based upon the fact that we all encounter the stem of flu all the time. So do these cells lack the ability to re-enter an active GC? Probably because they're not that mutated and their mutation rates remain the same. The lack of increased SHM over time. So AM1 and AM3 are associated with an ongoing and maturing response, so trafficking in and out of those germinal centers, increasing somatic hypermutation, associated with short and long-term antibody responses. And so can we extend these studies to naive B-cell targeting immunogens like the EOD GT8? And we're actually going to use some of these panels that we've developed to analyze that clinical trial that starts next month. So we're using this integrated technologies to provide additional immune monitoring capabilities. It may allow us to differentiate between pre-existing and recently generated memory B cells. We employed whole transcriptome sequencing to iteratively design phenotyping panels. And we continue to refine these parameters. We're not there yet, but we're getting there. And we're going to deploy this on the EOD GT8, because it would be very nice to look at something that is a neoantigen. We've seen flu all the time throughout our life, as I showed you. So it'd be very nice to look at which phenotypes we get when we use something that no one's been exposed to. So I'd like to thank a whole cast of characters that are involved in the EOD GT8 trial, all of these people. For the reagents of the EOD GT8, which is Dr. Bill Sheaf, those in the RICS Immunology Lab, those in the Structural Bioinformatics section, which is overseen by Peter Kwong, and also in my group here, particularly Sarah Andrews, Michael Chambers, and Alberto Kijiji. And thank you very much for your tremendous hospitality and attention, and I'll take any questions. Thank you.
Thank you, Adrian, for an excellent talk. Question, you show the differences across the global populations. I'm wondering, as I've been thinking a lot about aging in my talk, what about the age of the individuals? Were they all of the same age? As we've been looking at age-associated B-cell changes in vaccines, like the TBET positive CD11C cells recently with Mike Betts and the lead alter. So I'm wondering how those play in. And since those also seem to be, in some of our recent work, the atypical ABCs that you showed, the 21 negative, 27 positive, seem to be expanded in aging and associated with reduced flu vaccine responses in older, healthy individuals.
I'll start with your second point first and just say that I would love to be involved in some of the flu vaccine trials that we have, but sadly I'm getting too old to do it. Right, so our selection criteria for those individuals that are going into the clinical trials is really quite narrow. So I think it's between 18 and 50. On your first point, those cells that we obtained from Rwanda and Uganda and South Africa, they too were restricted because they were all recruited initially for clinical trials, so the same thing applies to them. So we're not looking at individuals here that are over the age of 55, 60, 70, where you would see a lot of that. And we have done those kind of studies in the past where we've looked at the distribution of different phenotypes just based on CD21 and CD27. And you do see there's a fluctuation within those. Like you get a lot more exhausted cells or atypical cells in those individuals that are getting older. So yes, it is a consideration. Now, whether microbiome affects this, which is what I'm kind of thinking, in the naive B-cell population, that's something we can talk about, because I think it's fascinating.
Yeah, thanks.
Yeah.
Nice job, Adrian. Getting back to your basic B-cell biology slide, it's my understanding that the decisions of which mutated B cells to let out into circulation and which ones get restricted and don't get out is made in the germinal center, and that is also related to restricting self-reactive antibodies from getting out in circulation and recognizing foreign antigens and let those B cells out. Maybe you could say a few words about that process and what the determinants are.
That's a very good point. I mean, there are a lot of checkpoints within the germinal center. But so if you have, rather like with T cells, if you have, if you're too reactive against a antigen that's presented on a follicular dendritic cell, then yes, it will go to apoptosis as well. So there's a lot of death going on there, a lot of death. And so but one of the questions that we're trying to ask is, what are the signals that are actually of the memory B cells? What are the phenotypes and what are the signals? And is there any difference between those phenotypes and signals that are present in germinal center memory B cells, which we can easily get hold of? In particular, let's go back to the EOD GT8 clinical trial. We have things called fine needle aspirates in that trial. And so we're going to be looking at this with a neoantigen, and we can probably tell, because we could do transcriptomics on those cells that are destined to die because they're self-reactive or they're not getting through checkpoint 1, on those cells that are destined to be memory B cells, and those cells that are destined to be plasma cells. So it's a great question, but there's not a lot known about the actual signals and the epigenetics that goes on within the GC in the selection.
So, Adrian, in follow-up to Alan's question, why is it so bad to be old? I mean, is it the miserable environment of inflammation that Alan is suggesting? Are there not enough naive B cells floating around to we lose some over time? Is the repertoire simply minimized in any way? Why are you so miserable as an old person?
Clearly not speaking for myself. But I would say that the ratio, and we noticed this in our experiments, the ratio between naive B cells and those memory or those that have countered antigen, so those that encounter increases, and it's not just the relative numbers, it's actually the numbers of naive B cells decreases as you get older. I don't know, I think we're just a bit tired recently, at that point. I think we, particularly in influenza, we encounter all these flus, and it's remarkable about the recall of the ones that we can make antibodies to. Maybe those, for example, in 2009 for the H1N1 pandemic, those individuals who were most affected were not the older population, because they have fantastic immune responses. They still have intact fantastic immune responses against those older H1N1s. Whereas the younger people who are still trying to catch up don't have those immune responses and can't protect themselves from the 2009 pandemic. And maybe if we go back and look at the epidemiology, the H3N2 infections that we've just had, that will show us that individuals who were exposed prior to the 2014 flu were more susceptible than those individuals exposed after. So yeah, I mean, we encounter so many different antigens, and I think just getting old, we reduce the output of our naive memory B cells.
So if there is no any more other questions, I would like to thank you again for your great talk.
Thank you very much.